Assalamualaikum everyone, welcome back to this channel. This is me Arya Wahid and you are on the channel of Medicare. So as you know, we have discussed about the antigens, we have discussed what are the properties of them, what are the types of them, we have classified them into the two classes. First we have discussed the major classes which are as self antigens and non-self antigens. Then we again subdivided these two classes, and again, the second classification of antigens is about the T cell dependent and T cell independent antigens. So, all of these kind of information has been discussed in the previous lecture. So, if you don't know about these kind of information, then you should definitely go and visit these channels, visit my channel, and also see these videos so that you can clearly get the information about that. But we have covered all the things, all the information about the antigens. Now our topic of discussion is about the antibodies. So you know that antigens help in the production of antibodies. So our main focus and our main protein which produced inside by our body is a antibody. So what is it? Let's discuss about that. So antibodies, they are the Y-shape proteins. So they have Y shape, as you can clearly see here. They have Y shape and they are proteins in nature. So they are made up of amino acids and they produced by the immune system in response to the antigens. So they are also called as immunoglobulins. So antigens are also called as immunogens, and immunoglobulins is the antibodies, so represented by Ig. So as you know, all the antigens are not immunogens, but all immunogens are antigens. So you should know about that. Like heptene is a antigen, but it not a immunogen because it is not producing the immune response. So this concept is discussed in the earlier lecture. So you should definitely leave see that videos if you don't know that concept. So now I have given the short introduction about the antibodies, that what are them, how their shape is looking like, and what they are doing inside your body. So let's discuss what is the structure of it. So which is the structure of antibody and how it is made. So actually it consists of four polypeptide chains, four polypeptide, not one, not two, it consists of four. How for? I will discuss it with you. Two identical heavy chains and two identical light chains. So as you can see here, this is, as I am drawing it, this is the heavy chain. This is a second heavy chain. One, two. This is a light chain, and this is other light chain. So this is one, two, three, and four. So there are four polypeptide chains here. And one thing you have to remember, there are two regions on the polypeptide chain. First is the variable region, which can also be called as a Fab region, and the second is the constant region, which called as FC region. So what is the variable region? It is also called as antigen binding site. So this region, okay, this is called as antigen binding site, while the constant region is called a receptor binding site. So here at that point, this is called as receptor binding point, and I have drawn the whole structure about it. So after discussing the structure of it, and now we are going to move and discuss about its functions. So the functions of antibodies. So first, they are helping in the neutralization of the toxin. They help to protect our body from the pathogens. So a lot of pathogens, a lot of toxins move into our body through air, through touching, through eating the food. So antibody is the natural agents which are produced, and they are protecting us from them, and also they will activate our immune system. And the last function of antibody, this is a new concept to you. Antibodies, one of the most important function of it is the apocytization. So what it is, let's discuss that. So it's mean increase in the phagocytosis of foreign particle by the immune cells. Means your immune cells which are already present in your body, like neutrophils, like basophils, like macrophages and monocytes, all of them. So they are the immune cells. So actually antibodies are activating them, are stimulating that some kind of foreign particle enter inside your body and you have to be activated out. So they are activating them, and they enhance the recognition and ingestion of antigens. So they allow your body to readily identify in what kind of region your antigen or your foreign particles are present, and then after the recognition they will go digest and phagocytosis of that antigen and completely kill it through your body. So that's why your antibodies are also called as opsonins. So that is the third in name of antibodies. So someone can ask you in a Viva question that what is the third name of antibody, that why what they can call it as instead of immunoglobulins. So you should know that opsonins is also a name of antibody, and why we give that name. Now you know the reason of it. So let's discuss the third important point about the antibodies, and this is about the antibody production, that how the antibodies are produced inside your body. So what is actually happening? We know that the antigen is responsible for the production of antibodies, but how? Actually, there are some mechanism of actions occurring inside your body which are responsible for it. So there are some steps which occur inside your body and through which the whole cascade is doing and performing this work. So how it is doing? So I have drawn here the four steps here. First step is the antigen presentation. Antigens presenting cells are present inside your body. They will attach themselves with the antigen. And after this attachment, they cause the phagocytosis and produce a complex which is called as major histocompatibility complex, abbreviated as MHC complex. Okay? After this complex formation, second step occur, which is the T cell activation. Now your T cells will activate it out. How? This MHC complex, major histocompatibility complex, it will combine itself with the T cell receptors. So these are the receptors present on the T cells. They will attach to them, they will bind them and activate them. And as a result, after the activation, the T cells will begin to release a chemical which is called as cytokinins. Now, the third step arrives, and this third step is about the activation of beta cells. So now your beta cells will activate, but how? So this cytokines which released by the T cells, it sends signals to the beta cells. As a result, the beta cell activate. It means the beta cells, it sends signals to beta cells, and as a result, the beta cells will identify that a particular antigen is present. Now it will begins to produce the antibody against that particular antigen. So first it will produce the plasma cells which convert themselves into the specific antibody, and also it's produce some memory cells for the memory. Like if again this antigen enters into your body, the memory cells are present, they will kill that antigen, so protect them from that disease for the second time. Now, the fourth and the last step is the agglutination. Agglutination means agglute to form the lumps. How? Actually, agglutination is a reaction in which your antigen and your antibodies combine together. I have explained to you in the previous slide that antibodies have a site which is called as antigen binding site. This is a variable region, FAB region. So on that region, they both of them combine and they form a lumps together. Lumps be coagulation. They form coagulations. They enhance the pathogens more recognizable. So now your pathogens will enhance themselves. They become more recognizable. As a result, they will detect, or they will be enhanced, or they will be recognized by your different kind of immune cells like immunogens, so sorry like they will macrophages, neutrophils. So after that the phagocytosis will occur, and now your antigen is killed, it completely removed off from your body. So this was all about that how the antibodies are produced, and what they are doing inside your body. So we have discussed about the introduction of antibody, about the structure of it, how its nature is what. We also have discussed its functions, and now we also covered how it is produced. The next topic of our discussion is about their classes. So how many classes of it? You know very well, there are five classes of antibodies. So let's discuss that. So now we are going to start the classification of antibodies. So they are classified on the basis of a name which is called as gamete. GAMED. So every word is indicating a particular antibody. Like G is indicating IgE, which is the one of the most abundant. Then there is IgA, which is the second most abundant antibody. Then it indicate IgM, which is the third most abundant antibody. Then it indicate D, which is the fourth important antibody. At the last, it indicates the IgE, which is the fourth last important antibody of your body. So, what is the IgE? It's again subdivided into four subtypes 1, 2, 3. They are divided on the base of their amino acid sequence, length of hing region, and disulfide bridge. So their structure is given, they are joined together with the J chain, and the structure is given and they are monomeric. Mono mean one, meric mean structure. Location, they are present in your blood, in your tissues, and one of the most important in the placenta, and having the abundance of about 75%. Their function is they can cross the placenta except the IgG2. It cannot cross the placenta, while others Ig1, Ig3, IgG4 they can cross the placenta. They play important role in the phagocytosis. They cause precipitation, neutralization, and also help to treat the chronic infections. They also help in the function of opionization. Mechanism of action is that they provide the immunity to the fetus by crossing the placenta, and they also triggers the complement system, lead to pathogenesis lysis, and increase the phagocytosis. IgA, it has two subtypes, 1 and 2. IgA1, IgA2. They are dimeric, their structure is given, and they have J chains. Their mucosal surface is present. They cause the secretions from the breast milk. They are present in the secretion of your saliva and also present in intestinal secretions, having the abundance of 10 to 15%. Their mechanism of action, they prevent the pathogenesis by attaching to the mucosal surface. They are present on the mucosal surface, they are present on mucosal sites like your respiratory system, and they prevent you from the infection causing agent like bacteria, salmonella, and vibrio. So these are the two bacteria's name. Function is the neutralization, mucosal immunity, barrier for pathogens, prevent infections of bacteria, and they are used as the first line of defense because they are present in your fluids. IgM, it has two subtypes having highest molecular weight. Present in pentameric structure, structure is given in the diagram having J chains in them. Present in the blood, IVF, intravascular fluid. Also present in the body fluids but not present in the secretions. Only the antibody which present in the secretions is the IgA. It about 6% abundant in the inside your body. Their function is the agglutination, complement system, primary immune responses, phagocytosis. They have a lot of sites through which antigen can bind, so therefore they have a lot of immune responses. They bind to multiple agents leading to pathogen lysis. Beta cells, from beta cells they produce in the monomeric form, but when they goes to blood they become pentameric form. IgD, having two subtypes, molecular weight is 18,000. Monomeric form, the diagram is given. They are present on the surface of beta cells and about 1% abundant in the body. Antigen recognition, it is helping in it. This causes the B cell activation and helping the antibody which is called as IgM. So this is the one of the most important antibody which is helping other. Next and last type of antibody is IgE. It has two subtypes, 1 and 2. Their structure is given. They are monomeric, present on the mast cells and basophils tissues. They are attaching to the mast cells, causing the release of histamine, prostaglandin, and neurokines, which cause increase in the inflammation. They are playing important role in the allergy, defense against the parasite, and mast cell activation. So this was all about the today's lecture. I hope that you will understand this lecture. If you like it, then please subscribe to my channel, like my videos, and also press the bell icon to get the notification of all the videos which I have uploaded. Thank you so much. If you don't subscribe to my channel, then go and press the subscribe button to get all the videos uh, for every topic. If you have still some confusion, then you definitely leave a comment for me. Take care. Allah Hafiz.